What is magnetogenetics? And the story begins with a couple of papers in about 2015. But the basic idea is that what if you could tag a magnetic particle and get it to bind to an interesting protein, in this case a membrane protein, which is a thermally sensitive one, and use it to switch that channel open or closed. Right? And this was basically reported in this paper. But the idea, I think you can see, is right here. If you look at the, is there a pointer somewhere?
It's right there on the ledge.
Oh, I'm blind. Thank you. Right. So the idea is to find a little magnetic nanoparticle, and this is one. And actually, the nanoparticle is one that's throughout our bodies. It's ferritin. Ferritin is the iron molecule that is used to store iron in the body. They played a nice little game by taking the iron molecule and gluing it onto this protein. And you can build a sort of a nice tagging system by which, in principle, you can inject this, have it dock to something on the molecule, and therefore you can stick it around. And then after you've done that, you apply either RF or a magnetic field, and you open up, in this case, a calcium channel to do that. So that's the idea. Quite a bit of work to show that it actually works. I don't want to spend a lot of time on this, and also partly because I don't understand enough biology to be able to describe this properly. But there are a bunch of things that you do. This is the one that you think will work. You can also get the ferritin to stick to the membrane, or you can just disperse it everywhere. This is just an assay. Probably you can't see it very well, but the point is because you've also got GFP and various other molecules stuck on here, you can check that the things are where they're supposed to be, and that sort of works. Then the next one was an experiment that they did with mice, so this was aimed to control blood glucose. So basically you inject a mouse, you wait for it to get expressed, and then in this case you microwave the mouse, gently microwave the mouse, I think, to do this, right? And what you can see, according to this, if you believe all of this, is that blood glucose in this case is red. So the one that you wanted to do, that you believe should work, is the green one. The blue is one of the controls where there is some ferritin around, but it's not necessarily linked to the channel protein, and this is the control. And done about half a dozen times. It's plausible at some level. But that was RF. This is essentially the same experiment now done, but with a magnetic field. And again, here it is. So you're on for a time of about 60 minutes in a magnetic field. And these are the two cases. So one of these things, well, they're basically both with the ferritin in, but it's sort of with and without the magnet. So that's all very nice. But then the question comes, okay? Yeah, maybe it works in practice, but does it work in theory? Right. So this is by the way, this is a quote typically about Chicago economists, right? That we do this. Because the kind of idea that's proposed is I have a little magnet in here. I'm applying a magnetic field. I can align the magnet to the magnetic field. It tugs along the molecule and moves it there. And no, that's a nice story to tell, but it's actually hard to make sense out of that. And that provoked a rant from Marcus Meister. Actually a rather useful paper, better say, that basically he just says, it's 5 to 10 orders of magnitude away from being a plausible mechanism for what's happening, and therefore throwing some doubt on all of that. And so, what's the critique, basically? I'll talk a little bit more about ferritin, the molecule itself, but actually ferritin is I should say, it's something that grows a nanocrystal of ferritin inside some package, sort of a 20-former holding all of this thing together, and you gradually sequester iron oxide in there until you build up a nanocrystal. The nanocrystals, when they're fully formed, are all pretty much identical. They're about 4,500 iron atoms, but they're not ferromagnetic. They're actually antiferromagnetic. So what does that mean is that there's a very strong magnetic interaction between the ion spins, but the ion spins are basically anti-parallel with each other. So I have an ordered state, but it's not a magnet. I mean, it's not. It turns out to be lightly magnetic because, of course, the crystal has a surface, and on the surface there are some uncompensated spins. So it turns out that the magnetic moment is about n to the 1/3, where n is the number of particles. So that's about 20 ion spins. That gives you 100 Bohr magnetons. And so if you take this number, compare it to kT, it's only about 20% even in 1 Tesla. Okay. Furthermore, if you start computing the force that you could get by taking this magnet and trying to orient it in the field, it's going to produce a force of the order of about 10 to the minus 23 newtons. So we've seen forces measured in this talk, piconewtons you're proud of, right? So that this is kind of a long way off from being really plausible. Now, something even worse than that is that actually ferritin is what's called a superparamagnet. So I said it was magnetically ordered, so the bulk material orders are around room temperature. But ferritin is so small that in this kind of paramagnet, it turns out that there are two equivalent states. I can have spin up, spin down, or spin down, spin up. And so there are two possible ground states, like a regular ferromagnet, where I could point the magnet up or the magnet down. And the system is small enough that it thermally fluctuates between those two states. It's called a superparamagnet. I'll show you some more about that. Okay. And at room temperature, it's actually fluctuating on a scale of nanoseconds. So there's no plausible way, again, that the little magnet stays pointing in the right direction for a persistent force due to a field. So that exercised Marcus and a few others. I particularly like his rant here, which is about this. He said that I'm not sure whether this is fair at all. I mean, I always tell people that if you've got experimental data, you should publish it. And you shouldn't have to have a theory in order to get it there. But as you say, he was because this paper and a couple of similar papers caused quite a fuss. There were review articles written all about this. And Marcus said, what's the community doing? As he says, claims that violate the known laws of physics often turn out to be wrong. Right. So does it, right? So I want to spend the last piece of the course saying, having the last piece of the talk saying, is there something plausible that could explain this? Assuming, of course, that the experiment's actually correct. I think last year, actually, there was another paper published by some of your colleagues, actually, right? With I think a good chance of being the right mechanism, although I think not expressed in exactly the same way. But in particular, they repeated some of these experiments, but I think under much more controlled lab conditions. And agree with it. But they also pointed out that there are other effects that can take place in a magnet, and in particular, that if you can change the entropy associated with the spins, then there's a lot more stuff to be had. So let me take you through that a little bit and a little bit more about the microscopic story. Okay, so as I said, ferritin is an ion storage molecule. It's in everything. It's pretty much deeply conserved. It's all the way from bacteria up to us. It has a protein shell, and it contains about a 7.5 nanometer core of something which is actually a well-known mineral, or a version of this. It's called ferrihydrite. If you ever see a red stuff leaching out of a sewer or something like that, assuming that it's actually the red is iron, it's typically this molecule. So it's rather like hematite, but hematite is just Fe2O3. Hematite doesn't have any water in it, and this has variable amounts of water, but this is kind of the official composition. Okay? But it's a pretty robust antiferromagnet as in bulk. So that means that the spins are all locked together in an antiferromagnetic configuration, and it's got a tiny ferromagnetic moment, but that's a finite size effect. So I said there's a mineral, we can do this. And then the other point to realize is that this order flips as a function of time. It's a superparamagnet. Okay. So let's see if we can get some handle on how much magnetism there really is in this material. It's not easy to do because, of course, if you've got nanoparticles, you can't put a squid on top of it and try and figure out what the magnetism is, especially if it's fluctuating. But one way of measuring this is a very old effect called the Canton-Mouton effect, measured in 1907, that just says that if the refractive index changes with the field, right, and the theory of this was done by Néel, but simply you expect this coefficient to depend on either the change in the susceptibility or the change in the magnetic moment. It doesn't matter either of each of these things. And this group, actually, for different reasons, studied nanoparticles of this material, and you can make it in lots of different kinds of ways, and that's sort of interesting. So actually, this is bacterial ferritin, actually, which is down here. Everything is sort of nicely quadratic in the field. These are other nanoparticles which also contain the same mineral, but they're made in different ways. I think some of these things are things that you buy over the counter to improve your iron and can swallow and drink, and so you see all of these effects. And on the basis of that, you can use this to determine either the susceptibility or the moment. And in particular, you can also look at what you would expect if the effect is not entirely linear. And the one where you see it the largest is an iron sucrose solution here. And in that case, you can actually get fields which are big enough to saturate the birefringence, which means that you really are lining everything up. You're a very long way away from doing that in this material. I think it's not really understood why it is that there's much more moment on these particles than there is in ferritin. Now, the next thing is that this isn't the first controversy involving ferritin. I said the spins flip up and down. Okay. Of course, that's an activated process, so as you go to lower and lower temperatures, you'd expect it to stop. Well, it doesn't. It actually keeps going. This was proposed back, actually, in the 90s, that what you were seeing at very low temperatures is actually quantum tunneling of the moment. I don't want to go further on this, but it meant that the topic got well looked at by this. And actually, this remains pretty controversial about whether this was really happening or not.